Okay, we now turn our attention to the endocrine system, chapters 18 and 19. It's kind of artificial to separate them into completely separate central and peripheral endocrine tissues because some of our neurohormones secreted by some of our more central endocrine glands can promote secretion from other distal endocrine glands in the periphery. And so I'm going to collapse this lecture into all kind of mixed chapter 18 and 19. Here, seen in figure 18-1, are what we typically consider as endocrine tissues. But as an important reminder that many tissues secrete hormones that are not prototypical endocrine tissues. We recently, for example, talked about leptin, a hormone secreted from adipose cells that influences our appetite. But adipose cells are not shown on this figure. Muscle cells secrete irisin and interleukin 6, and those also are not shown on here. Irisin might influence the relative brownness of adipose tissue, which tends to have higher metabolic cost than white adipose tissue. These are the typical endocrine glands. Now, a reminder that some of these, of course, have other functions. The heart is not just an endocrine organ. It is an organ that is critical for imparting some force onto blood to promote a blood pressure, which allows for delivery of blood. But it happens to secrete atrial and brain natriuretic peptide from the atria and ventricles, respectively, and thus is also an endocrine tissue. So a couple of things we said previously about endocrine tissues is that their specificity arises from what tissues have responsiveness to the hormone, so have receptors. So a hormone will go into the circulation and has the potential to influence any cell that happens to have receptors to it. This is different from our nervous system in that the nervous system its specificity of what it's going to affect is partly related to where it synapses, with what tissues it synapses. Now, the nervous system signals are much faster by comparison to endocrine, and the nervous system signals are not as long-acting as the endocrine system signals, as these hormones will stay in the circulation often for some period of time. In some cases, neurons are secreted, hormones are secreted from neurons. And when that happens, we call those neuroendocrine or neurohormonal communication. So, vasopressin, oxytocin are examples where they are secreted from neurons into the circulation and can act on target tissues. In the case of vasopressin, this might be cells of the distal and collecting tubules, and it would result in an increase in aquaporins embedded in the membrane on a cyclic AMP related second messenger pathway. So this is figure 4-19 in your text. Some basics of different aspects of endocrine communication are covered in chapter 4, and as a brief review, we have hydrophilic hormones, which are peptide hormones where we have an awful lot of those. Some examples would be growth hormone, insulin-like growth factor 1, prolactin would all be examples of peptide hormones. We have catecholamines, which are hydrophilic, and they're derived from the amino acid tyrosine, and I'll show you some pictures of these in a moment. And in these cases, because they're hydrophilic, they do not need to be bound to a protein because they are soluble. They're hydrophilic. Some hormones, like growth hormone, can be free, but it can also bind with a growth hormone binding protein in the circulation. And insulin-like growth factor 1 can bind with one of 6 IGF-1 binding proteins. But they don't require being bound. Some of these may be bound, and they, in both cases, are going to be binding with a receptor on the surface of the target cells. So hydrophilic are going to act via a surface receptor. Whereas lipophilic or hydrophobic hormones, like thyroid hormones, which is also derived from tyrosine, and steroids, which are derived from cholesterol. These, because they're not water soluble, will have to be bound to plasma proteins, and they are going to bind with a receptor inside the target cell because, being lipophilic, they can go directly through the phospholipid bilayer. So this is an example of a peptide hormone, and in fact the peptide shown here is insulin. You could see it is a long chain of amino acids, and each amino acid is abbreviated on this figure. And after this long protein is made, peptide is made, that's not actually that long. This, what is called the C-peptide, is cleaved off after a couple of disulfide linkages form between some adjacent cysteine residues, forming cysteine. And as a result, what we would get is a mature insulin molecule, which is this A chain and B chain without the C peptide. And so once we make this string of amino acids, making a peptide hormone, it may still need to be processed before it's in its mature form. In fact, when this is just one long chain, it's called pre-proinsulin. And then after the disulfide linkages form, which is called proinsulin, and then after the C-peptide is broken off, then it is called simply insulin. This is showing you catecholamines of epinephrine, norepinephrine, and dopamine, and they are all made from the amino acid tyrosine. You do not need to know the chemical structure, but you should know that they are derived from the amino acid tyrosine. Whereas our steroid hormones are all made from cholesterol. So these features look a lot like cholesterol, and you can actually see here various intermediate steps in making estrogens, testosterone, progesterone. A couple here are underlined, which have historically been somewhat common supplements that people might take to try and enhance the amount of testosterone. But based on the enzymology that causes these transfers, it also usually increases the amount of estrogens as well. And so while taking such supplements, you may not only enhance the testosterone, but the amount of estrogens and some female secondary sex characteristics. A brief description of some of the intermediate steps so that you can understand how these different molecules are interrelated. Cholesterol can be made into by sequential steps, ultimately into progesterone and aldosterone through one kind of part of this tree, and into cortisol and another, glucocorticoid, similar to dexamethasone, which has recently been reported to be useful in reducing deaths in COVID-19 patients. Here's that dehydroepiandrosterone, which is a precursor to androstenedione, which can be used to make testosterone, but some of it also making estrogens. So we'll come back to this figure later. So hydrophilic and hydrophobic hormones. Remember, hydrophilic means they're water soluble and they can be transported free. They may sometimes, in special circumstances, be bound to proteins. They're going to bind with a surface receptor, and that will activate a second messenger system. Whereas hydrophobic hormones or lipophilic hormones will circulate bound to plasma proteins, but will act via receptors in the nucleus and influence gene regulation.